Alright, so if you're using a hard device, then go ahead and use that hard device for the whole time. If you're using a cell phone, then you need to plug in that to the session ID. Do not switch between a cell phone use and a clicker, a hard clicker. It won't work. Okay, we cannot match the data set. One or the other, please. So if you could go ahead and answer that first question. And we'll come to the answer here in a moment. I want to orient you to what we're doing this week and give you an analogy. We're going to be purifying RGFP from bacteria this week, and it's going to take us several weeks to go through the entire analysis of that. When we break open those cells, about how many genes are present in E. coli? Somebody throw out a number?
280.
380, that's kind of low. 380, that's probably kind of low. It's in the thousands. About how many thousand genes are in E. coli? Somebody want to toss out another number? 3,800. 3,800? Yeah, about 4,000. Close to. About the 4,000 or so. It depends upon which E. coli strain. And if we're interested in one particular protein, we're going to add a tag to that. Okay, so an example of that might be if I've got a check in my pocket for $5,000 and I need to give that to the lucky student of the campus. There are 25,000 students here. How do I make sure I get the check to the lucky student? Good. Something unique about that student. The student ID. What else would be another unique item? I'm sorry, what? No, not good. Not picture ID, not good because I got a twin. Doppelganger. What's another unique identifier that you have? Fingerprints, good. What else? I'm sorry, what?
Tattoo.
Yeah, maybe. Dental records, social security number. I mean, these are all unique, yes, DNA, but certain regions of the DNA are very same for everybody. So it's got to be a unique identifier. We're interested in purifying RGFP, correct? Recombinant green fluorescent protein. What's the unique identifier that we're going to use to purify it in relationship to, there's 4,000 different proteins in a cell. What's the unique identifier we're going to use? The His6 tag. We could also use the express epitope tag. That's very unique too, but we're going to use the His6 tag. And so that's what we're going to be working through today. Before I go on to the next slide, make sure you clicked into this. Let me go ahead and close it out. All right, going once, twice. There's got to be more than 92. Going once, twice. So good. Most of you got that correct. The increased turbidity, the cloudiness preserves to more bacteria. So we've got this pool. We've got this pool of proteins, and we need to identify a particular protein very rapidly. It's like trying to identify Waldo. Okay, can be kind of hard. Okay, and so we'll kind of refer back to that towards the end of class today. So, looking for Waldo, there's a unique identifier, and I'll let you figure that out on your own here shortly. But our unique identifier is the His6 tag. And so today we're going to talk about that purification procedure. So this is the entire series of experiments that we're doing in this next 4 or 5 weeks. Experiments 3 through 8. And so I've already done 3. The important thing with experiment 3 is that you now know what a G culture looks like. What does the G in a G culture stand for? not growth, it stands for GFP. It's the strain, it's the bacterial strain that has the GFP gene present in it. And the V strain, what is the V strain? It's the vector control. It's the exact same identical strain except that it lacks the gene for GFP. And so, experiment 3, we learned about that. We saw what fluorescence looks like in the presence or absence of IPTG. We saw it on plates, and we saw it in liquid cultures. This week, so there's experiment 3. Experiment 4, 5, 6, 7, and 8 are all going to work together, so you're going to be writing this combined lab report, and the entire purpose of this series of experiments is to see if we can express. Can we express GFP? Can we? Yes, we already know that. We did that in lab 3. You saw that on plates with and without IPTG. And you also saw the controls on that. This week we're going to look at purifying that GFP. Purifying a His6 tag form of that GFP, that recombinant green fluorescent protein. And we're going to be doing that from a species called Escherichia coli. So there's a species name, just like Homo sapien is a species name. But every one of us in here, well, maybe not me, but everyone in here is a Homo sapien. Okay, I've been called other things, but regardless, I also each and every one of you have a specific name, and the specific name of the E. coli strain we're using is called VL21DE3 PYSS PRSET AGFPUV. That's a specific species. That's a name of a strain of a species. So the analogy, your name, and the species analogy is Homo sapien. And you're going to be using nickel-agarose affinity chromatography techniques to do that purification process. So that's what we're doing this week. This is what we did last week before you came into class. This is how you're going to be starting your combined lab report from the perspective that you actually expressed RGFP and E. coli. That you started with a G sample. And it's G0, 0 indicating 0 minutes prior to addition of IPTG. After we took that G culture, it was growing, we added IPTG, and we grew it 3 more hours at 37 degrees Celsius with shaking. And as soon as we get done with that, then we have a G3 sample. That's the G culture, 3 hours growth post IPTG induction. And then there's 220 students in the course and 10 lab sections. We made 230 pellets. So I had a TA take 15 mil culture, 15 mils out of the culture, 230 times. They spun that down, they got a supernatant and a pellet, and they decanted the supernatant. The supernatant was the leftover Luria broth, or LB. And the end result is what we call a G3 15 mil pellet. That is to say, it's a pellet of bacteria. That pellet of bacteria comes from a volume of culture that started at 15 mils, and it had been induced for 3 hours. Okay? You're going to come to class this week and get that G3 15 mil pellet. This process takes about, it can easily take 14 to 16 hours to do. So that's why you didn't do that. That's why we're giving you the pellet. But when you write your combined lab report, you write it from the perspective that you did this procedure. The technical stuff is written in your lab 4 lab protocol. Lab 4, you're going to take that 15 mil pellet and you're going to go through what's called a freeze thaw method to create crude extract. We'll talk about that today. And the end result of that freeze thaw method is to create something called GCE. GCE stands for that crude extract from the G culture. Okay. Then you're going to pass that over a nickel-agarose affinity chromatography. That GCE gets put on there. We're going to develop that column with a wash buffer and elution. And we're going to collect a multitude of washes and elutions. The ones that are important for the lecture are going to be washes 1 through wash 6 and elutions 1 through 6. Okay? And you'll notice that there's more fluorescence in the elutions than there is the washes. When we come back from next week, we'll go into lab 4, or lab 5, and we'll do a Bradford analysis to determine the amount of protein that's present in our fractions. That's total protein. And then to determine, knowing the volume, we can calculate the concentration of our particular fractions. And again, these are the important fractions that we're going to be saving. Then we're going to go into lab 6. We're going to do an STS Page gel. And we may or may not do a Kamasi blue staining gel. This is to estimate protein purity and relative molecular weight. We just spent about $60,000 on some new equipment, and y'all get to be the guinea pigs with that new equipment. And so we'll know Friday. We've got a rep coming in to take a look at and help us through that new equipment. You may not be doing Kamasi blue staining, but the end result is you're going to see a figure that looks just like this, or very similar to that. Okay? Then you're going to have your exam, and then we'll come back and do lab 7 and 8. In lab 7 and 8, we'll do a Western transfer, and then another STS Page, and a Western transfer, and we'll confirm the presence of RGFPs and antibodies. So with that in mind, let's go ahead and see if you can click in again to that. What is the purpose of this series of experiments? And if you'll notice, the series of experiments comes from basically Martin Chaffee's Nobel Prize-winning science paper. Going once, going twice, sold. I think it's sold. There he goes. Good. All right, good. Y'all did much better than the Monday group. That's good. Y'all must be getting the answers from the Monday group. All right. So there are 4 intellectual steps to optimizing any protein purification scheme. And we're going to kind of talk about these today. Every instructor, every biologist, every scientist is going to go through these. And sometimes it's very rapid. It can take a matter of about 5 minutes, and they've figured out this stuff based upon what they're purifying. First thing is to develop a suitable assay procedure for your protein of interest or your molecule of interest. In our case, it's a protein. And we've got a great assay procedure. We use a light that's called an excitation wavelength, 395 nanometers. We shine that onto a sample. If that sample picks up that light energy and fluoresces it, in this case, in our case, if it picks it up and fluoresces it at a lower energy level, higher wavelength, it emits at 510 nanometers. Our eyes see that as green. Okay? We've got 2 ways of measuring that. We've got the way my daughter likes to look at it. She looks at it with a handheld UV light and she goes, "Ooh, this is really bright pink." It's not. I don't always use GFP. Sometimes I use different fluorescent markers. Other times she goes, "Oh, this is really dull. This is paisley." Okay, those are very qualitative measurements: low, high, medium, etc. You've got your quantitative. We learned how to do that using the machine, that spectrophotometer that measures fluorescence and reports that in numbers with a unit of relative fluorescence units. In this case, 100 versus 5,000, that's a 50-fold difference. That's a huge difference in activity. And so we did that in lab 3. Today we're going to talk a little bit about selection of the best source material. And that's our source material is going to be E. coli. But how do scientists view that? How do they look at that? Where do they go for their material? We're really not going to talk, I'll talk about solubilization of the desired protein right now. It's real easy. I don't have any slides for it. We prefer to purify proteins that are already soluble inside the cell. It's harder to purify proteins that are, say, found in the lipid bilayer membrane, because as soon as you remove them from that lipid bilayer membrane, they begin to denature. And so when they start denaturing, you're going to lose their activity. So most biochemists initially are going to be working with proteins that are found soluble in the cytoplasm, and that's what we're going to be working with. GFP is very soluble in cytoplasm. And then we're going to talk about developing a suitable series of fractionation steps, and I'll give a demo on how we go about that process of fractionating and what happens. And when we refer to that, we're typically talking about going from one column to another column to another column. Okay? Not the same column, but different properties, and we'll talk about that. So let's look at selection of the best source material. Let's say I'm trying to analyze a protein that's involved in Alzheimer's. So I can go for primary tissue. So what do I mean by primary tissue for an Alzheimer's protein purification? What do I need? I need some brain tissue. So any of you guys want to donate some of that? No. So it can be hard, but you say, hold on, no. I'm a donor. My ID says I'm an organ donor, so you can take my brain afterwards. Well, that's great. Do you have Alzheimer's? So it can be hard to get some primary tissue, plus getting the right primary tissue. And then you start working and getting tissues from some maybe older people that agree to that. They may or may not have Alzheimer's. So getting primary tissue can be difficult. But if we can get a little bit of it, then we can put it in tissue culture and we can begin to grow it and purify the protein away from the cells that we're growing. What's the problem of trying to grow primary tissue in tissue culture in Petri dishes? Does anybody have tissue culture experience? Read about that? Yeah, so what's the hard part about doing that?
It grows really slow and it usually gets contaminated.
It grows very slow, it can become contaminated, and if it's primary tissue, does primary tissue survive forever? No, it's got senescence in there. It's going to die at some point in time, typically 30 or 40 doublings. So whenever we do tissue culture, we're probably working with cells that grow that are immortal. What's wrong? What's an example of an immortal cell? Cancer. So if we're trying to purify a protein to talk out of a normal cell, but we're purifying it from a cancer cell, well, maybe that protein is not the right type of protein. Maybe it's not behaving properly. It's from a cancerous cell. So you got to think about those. And to be quite honest, it's extremely expensive. So primary tissue and tissue culture have some inherent properties in them that are great in the sense that, hey, we're getting the wild type protein, the protein from the organism we're interested in. But sometimes we can't get the primary tissue. It's too difficult. We can't grow it in tissue culture. If we're growing it in tissue culture, it's a cancerous form. There's complications to that. You have to, as a researcher, you have to acknowledge those limitations. Okay? The other three systems are yeast expression systems, bacterial expression systems, and in vitro expression systems. In order to use any one of those systems to purify, say, a protein involved with Alzheimer's, what do I have to have? Think back to Martin Chalky, his paper in Science. What did he have to have in order to express GFP in E. coli or in C. elegans? He had to have the clone from Douglas Prasher. He had to have the gene. So these types of expression systems require a cloned gene. Without the cloned gene, you're not going to be able to purify any proteins. And of those three in vitro expression systems, those are test tube systems. They're typically not done nowadays. They're very rarely done. They're specialty cases. I've only done an in vitro expression system once, and in all my readings, I very rarely see it, maybe once or twice out of maybe 1,000 papers. So most of the time, we see things in yeast expression or bacterial expression systems. And so that's kind of the new starting point for biologists nowadays as biochemists go to purify a protein. Whether we're purifying it so that we can, say, insulin, so that we can inject it into a human, so that they can fight off diabetes and so forth, or if it's purifying a protein so we can do crystallography studies, we'll use yeast or bacterial expression systems. And there are some advantages and disadvantages that, some of the advantages, it's extremely inexpensive. relatively cheap to do this. Minor space requirements. I could probably, if I had the right strains, I could probably make enough anthrax to kill everybody in the world out of a semi-truck, okay, for maybe $10,000. In reality sense, that's a small volume of space and a small amount of money. Okay? So typically much cheaper than, say, in vitro or primary tissue culture. It can create large yields. And what's nice is we can control the expression. So there's two types of controls of expression. One of them is constituents. What is constitutive expression? What's constitutive expression? It's always on. Constitutive. It's always on. We're not controlling it. And the other is inducible. And so we're using an inducible system. Our inducible system is the T7 RNA polymerase system found in the BL21 strain. So we're using the T7 system. There are other inducible systems like arabinose inducible systems that you'll go back and you'll review from your exam. There are lots of different inducible systems. What's nice about those is sometimes a protein is being expressed in bacteria or yeast is toxic to that bacteria or yeast. And so if we have it on a constitutive expression, that yeast and bacteria, they just die. They don't grow at all. And so what we can do is we can grow a whole bunch of bacteria or a whole bunch of yeast. So we have a very turbid culture. And we notice as soon as we induce it, it's going to make protein, but very rapidly those cells will die. And so we can get a lot of protein by simply growing a lot of bacteria and then at the last minute inducing it, letting it grow, and it'll die out because you induce it so heavily, and then we've got a lot of protein present in there. Not because each cell has made a lot of protein, but because we have a lot of protein, a lot of cells present. And that's one of the nice things about an inducible system. There are some disadvantages. One of them is post-translational modifications. There are things that are done to proteins after they come off the ribosome in translation. Things like methylation, sumylation, farnelization, phosphorylation, post-translational, very specific post-translational modifications where maybe an amino acid is altered or that polypeptide is cleaved in two. If that's what happens in the human system, but we turn around and take that gene and express it in yeast, it may not happen. It might, because yeast is a eukaryote, and so are humans. However, humans are multicellular and yeast are unicellular. And if we go to a bacterial system, there may be a whole bunch of different post-translational modifications done that may or may not occur on your protein of interest. So just because you're able to purify it doesn't mean that it's the native active form. There may be some of these post-translational modifications that are not done. Sometimes the proteins are not stable or they're not soluble. They fall out in precipitants like inclusion bodies. That's a precipitant. And the only way, and then also sometimes we might get small yields. And we say, hold on now. We've got this stability, solubility, small yield, post-translational modifications. What's going on there? Well, how do I know my protein is going to be active? How do I know it's not going to be insoluble? How do I know if I'm going to get a lot? You just have to try it out. Okay, you just have to try it out. And then, so there are some disadvantages to this, but most of the time we're going to be using yeast and bacterial expression systems. And once we are expressing them in yeast and bacteria, we have to have a way of breaking them open. We'll talk about that on the next slide. But in the meantime, there's got to be some type of purification method. And so we used to use traditional methods. And we'll talk about some of those traditional methods today. But nowadays, almost always, biochemists use tag methods. The tag that we're using is the His6 tag. There are other tags. There's an express epitope tag. We actually have one of those. There's a tag called a flag tag, like a FLAG, flag tag. There's a tag called a MYC tag, MYC tag. GFP can be a tag. It's a very big tag. Most of the time when we think of tags, we're thinking of something small. And so we'll kind of go over traditional methods today also. So as we've got this new starting point, bacterial expression or yeast expression systems, we've got a broad way we go about purifying. We've got to prepare that crude extract. And so to do that, we could use grinding in a mortar and pestle. And we've already done that. We did that in experiment 2 with alfalfa. You can use a blender, as long as it's not the same one you use for the Friday margaritas. When you use a blender, if you're using sand, you're probably going to be breaking open E. coli. If you're using glass beads, you're probably going to be breaking open yeast. I've done both of those, the blender and the mortar pestle. High pressure, a French press. I've seen that done. It's kind of messy. They just take a large volume and they press it through a very small opening, and it just basically busts open the cells. But it's not done very often because of the equipment. Sonication, using sound waves. I've done that before, and you saw Martin Chaffee did that in his experiment in the science paper. He used sonication or sound waves. And you can freeze thaw with liquid nitrogen. That's typically only done with animal cells, though. It's something that doesn't have a cell wall. And so these are all physical methods that we've either seen or already done or read about. The other way is enzymatic methods. And so we can add lysozyme to the breaking buffer. I've done that with yeast before. It weakens the cell wall of yeast, and then you can put it under osmotic shock and break open the yeast. We also can use lysozyme that's being expressed from a plasmid, and the common plasmid that's used in this is the P-lysess plasmid. Once you get done breaking and creating a crude extract, you typically, if you're trying to purify a protein, you're going to have to go through some additional steps. If you're just trying to look to see if you have activity, then you don't need to go any further. An example, experiment 2. We broke open alfalfa plants, and we looked for, in the crude extract, we looked for the activity of the phosphates. If you're trying to purify a protein further than just looking for its activity, you've typically got to go through these things to get rid of small molecules and to decrease the volume. And at some point in time, you're going to go through a series of chromatography steps. Normally it starts with an ion exchange, followed by affinity chromatography, then gel filtration. We are going to skip a lot of that. We're just going to go for one trial. We're going to go straight into affinity chromatography. And that's actually fairly normal when you have a His6 tag species, that you skip a column step. Or maybe you reverse the order of the column steps. And finally, you store it in -70 for further use. The time that it takes from preparing the crude extract to getting it into the glycerol storage buffer, that's very critical. The longer your protein is away from the primary source, then the more activity is going to be lost over time. And so we want to go through these fairly rapidly. We don't have to worry with GFP because we know it's a very stable protein, and that's why we use that in this course. So I want to remind you about, this was a slide of the bacteria here, bacterial cell wall and cell membrane, and this is after IPTG induction. And one of the proteins that we're dealing with, there's 2 primary proteins that we got to think about, at least out of those 4,000, in reference to preparing that crude extract and purifying. First is GFP, that's our target protein. The other protein we need to think about in that crude extract is lysozyme. That lysozyme is being made off of the P-lysess plasmid, and that lysozyme protein has 2 functions. The function that we do know is to prevent the leaky expression of the T7 RNA polymerase in the absence of IPTG. That's those thin arrows. But the thick arrows show what happens when you add IPTG. You get overproduction of the T7 RNA polymerase, and there's not enough lysozyme to prevent that leaky expression, and so you get our target protein production. The other thing the lysozyme protein is involved in, 2 things, is degrading the cell wall. Students always say, well, gosh, if you're making lysozyme and it's inside the cell, why doesn't the cell break open? Well, the cells are sick, okay? But the lysozyme interacts preferentially from the outside and the inside. So these cells are sick, but you've got to get some of them lysed open initially so that the lysozyme that's in those cells damages the neighboring cells. So this is an example of this in this process. And let me start. You got this we take our cells, we slowly freeze them, and these ice crystals form. They're going to puncture some of the cell walls and cell membranes. They behave like molecular knives. Now, in your lab reports and all, don't describe this as molecular knives, please. No, it's not really true. But the ice crystals will cause punctures, and when you thaw that sample, then the cell that burst open right there is going to spill its guts. Okay, so I've circled that in the center. Okay, and that osmotic shock causes that cell to burst open. It spills the cytoplasm out. 4,000 different proteins along with all the lipids, carbohydrates, nucleic acids, everything. Our target protein, GFP, and one of the proteins it spills out is that lysozyme. That lysozyme will then degrade the neighboring cells, which degrade the neighboring cells, and you get a chain reaction. Okay, and let's see. Here's a visual representation of probably what's happening in the I've got to find this next series. So there's your cells there. One breaks, and all of a sudden it just starts chain reacting there. Okay? And so each one of those squares is you can think of it as a cell, and once one goes, you've got that chain reaction. What you'll notice in this process, and it's hard to tell, but there's a couple in here that didn't break open. And what I want you to recognize is that not all of this, throughout biology, nothing's 100% efficient. It's not analog, it's not math. 1 plus 2 is always going to be 3, okay, by definition. But in biology, 1 plus 2 today is 2.9, and tomorrow it's going to be 3.1. There's fluctuations. Notice that we're not getting 100% cell lysing, so there's going to be some cells that are unopened. So we've got to purify that crude extract. We've got to purify our target protein away from that crude extract, away from those contaminants. And the traditional methods, we're going to be exploiting physical differences between your molecule of interest and all other molecules. So in our case, we're going to be referring to proteins, but if you're in chemistry, you're still doing the same thing. You're looking for physical differences between your chemical of interest and all the others. And so chromatography is going to have both a mobile phase and a stationary phase. That stationary phase is a matrix. It's the material in the column. It's the inner bed that's going to interact, going to have some kind of interaction with the mobile phase. And you're going to be exploiting differences in molecular binding that include things like the shape of the protein, the charge of the protein, positive or negative, or the amount of positive or negative, or the location of those charges in reference to the shape. Typically, we're thinking about affinity chromatography in that case. The hydrophobicity, how oily it is. Does it prefer to be in water or in an organic solvent? typically hydrophobicity chromatography is going to deal a lot more with organic chemistry than it will necessarily with biology or biochemistry. Or we're going to look for physical differences in size. And so the larger it gets, the higher the molecular weight. So we'll kind of use those terms interchangeably, size and molecular weight. So those are the principles behind any purification step. And as we do that, we're going to have these three balls that we're always going to be juggling at any one point in time. The first is purity. Every step we do, our goal is to make that compound more pure. And so normally we express it as a percentage. So if I say something is 90% pure, then that means 10% of the remaining compounds in there are contaminants. Okay? Then we're always worried about yield, the amount of that protein, whether it's the amount of your protein of interest or the amount of all the protein that's present. Okay? And so under certain conditions, like a Bradford assay will tell us the total amount of protein. An STS page gel will tell us, with some additional information, we could get our protein of interest amount. Normally we express this as an amount, as a weight, micrograms, milligrams, kilograms. Okay? Very rarely do we express it as a percent yield. Percent yields typically are used in chemistry purifications. Okay? And then finally, the activity of our protein of interest. Just because you have that protein doesn't mean it's active. It could be inactive because of maybe two things. Maybe it's denatured, or maybe the post-translational modifications are not proper. And so yes, it's three-dimensionally fairly close to wild type, but because it's not modified properly, it's not active. So these three things, every time we're working with proteins, we're always juggling and trying to prevent lost yield, lost activity, and trying to increase purity. So I'd like to do a demo, a dry demo on this concept of purity and activity. So you've got a table in there that you can fill out in your notes as we go through this. So here's a crude extract after I've broken open the cells, and you can tell that there's lots of different proteins in there. They're all pretty similar shape, but we've got a particular target protein we're interested in. The pink-haired, red-hatted glass person there, okay? And so that's our target there. And our first step in this purification procedure is that crude extract. You can see we've got, if you went through and counted it, there's 112 total proteins present there. And if you look through closely, you'll see that there's 8 of them are our target. So the purity is only about 7%. You know, when we did the alfalfa extract, that was fine. We didn't need it to be 100% because our goal was to see if there was phosphatase activity in alfalfa plants and what were the optimal pH and temperatures of that activity. So it didn't matter if there were contaminants. But in our case, our target, we want to purify that protein because maybe we want to crystallize it. We want to see its three-dimensional crystal structure. So we're going to add a column in here. This is a series of purification steps that we're doing. And so the first one is we're going to fractionation based upon pink hair. We have a special column that can distinguish the difference between blue and pink hair. And so we do that, and there's our result. So you see, I got rid of some contaminants there, all right? Now also, if you look in the upper left, ooh, what happened there? Was that a target? Yeah, it was a target. I lost it, okay? Not every step is 100% efficient. Sometimes I'm going to lose my target in that process. Plus, look, I got some other ones in there. I got those blue ones in there. I don't want those. Those are definitely contaminants based upon this column here. But remember, not every step is 100% efficient. Maybe the shape of that hair makes it look like that it's very similar to the shape on the pink. Okay? But regardless, those contaminants happen to get through. And so on this first step here, if I look at that, my total amount after that first fractionation is 58. The target has dropped to 7, and my yields increased 12%. Not very good, not a very efficient method. But we're gonna add it, we'll use it. We may choose, when we get done with this, to change this system and just get rid of that column and not even do that. Next thing, let's separate based upon hats, red hats. And so I go through and I've got a special column. I take this sample that you see on the board, I put it on top of a column that binds to red hats. And here's my end result. You see, upper left there, there's a contaminant there. It's got pink hair. She doesn't have a red hat. I don't know what happened there, but it wasn't 100% efficient. You see, well, I don't want this guy down here. Why is he here? He has a red hat. So the column worked for that particular fractionation step. And oh, lo and behold, right there in the center, I lost one of my targets. Okay? So not every, oh, what about this one here? This one right here. What's going on there? Why did we get it? It's not a red hat. Hat looks kind of similar, right? Triangle. Maybe it's just not 100% efficient. Guess what? It's not 100% efficient. It's definitely not 1 plus 2 is 3. It's 2.9. Okay? So, if you look at, you count 16, 6 targets, and now, ooh, that's a pretty good jump, 37%. That's a pretty good purification there. That's definitely better than the crude extract. I may decide to stop. But if I'm going to purify a protein for crystallography studies, how pure do I need to have that protein? Okay? Very, like 100%. Because if you've got particulate matter of proteins that are not part of your protein of interest, and you're trying to crystallize them, a crystal won't form. Or you'll get a crystal that is not a very good crystal. You can't get a good structure on it. So we want to get 100% pure. So let's go ahead and use the distinguishing physical feature of having or not having glasses. Okay, so we're going to separate based upon glasses. Now, these are all physical differences between those proteins. This is an analogy. We've got shape, charge, hydrophobicity, size. These are all various types of methods that we could use in biology. So look, got rid of some, got rid of a lot. And in the process, I lost another one of my targets. And why do I keep getting him? Okay, or the blue-haired person there. Well, the column did the right thing. It collected anything that had glasses. It collected anything that had a red hat. So that's a protein that has a very similar nature to our target. And it just so happens on the first step, it wasn't very 100% efficient. So now we look at this and wow, we're down to 7 total, 5 targets, and we're at 71%. Probably not good enough for crystallography studies, and may or may not be good enough to inject into humans if you're trying to purify a protein that's going to be an active biological ingredient that goes into some type of drug. So we'd have to go back to the drawing board and get rid of things or change fractionation steps. So there's the numbers if you didn't already get those. And I'm sure somebody next to you did. So let's move on. So let's take a look at this question, considering that third fractionation step. Going once, going twice. All right, good. Let's look at that for a second. increased, but the yield did not increase. The yield dropped from a total of 8 down to, I think, what, 6 or 5? So, during a protein purification scheme, typically, this is typically, every time you're maneuvering something, so if I get this front row, all empty glasses, and I pour milk into this first glass, these are all identical glasses, and she pours the milk into this glass, and it just goes down the chain. If I started at 12 ounces down at that end, what am I going to have down here? Less than 12 ounces, okay? Because there's residual stuck in there, and that's going to be lost activity, purity, or yield. Lost yield, okay? And so typically yield will decrease, so we've got to do whatever we can to increase the yield. And that could be using a spatula to get every drop out, okay? Or it could be get rid of some of the steps in between. There's a variety of things. Or it could be, maybe I want 12 ounces down there at the very end, and so I'd start with 13-ounce glasses. And I'd pour 13-ounce glasses, and I'll get lost yield along the way, and I'll at least have 12 ounces at the very end. Okay, so there's a variety of ways that you can increase yield, or at least prevent decreasing yield. Okay? Purity, hopefully it increases. If your purity doesn't increase, you've got a problem. Okay, maybe you should be in computer science. Oh, we're in the computer science building. So that's the goal. If your purity is not increasing, there's an issue. And hopefully, our activity is going to decrease along the way. It just happens. Our goal is not to have it happen. So we keep things on ice. We keep things in the freezer. We keep things in the refrigerator. We add protease inhibitors that prevent proteases from cleaving our proteins. But typically activity is going to decrease. I want to introduce you to another term that you're going to see in Lab 5, but I want you to see it now because we're talking about activity. Now, in that previous demo with the blue hair and the pink hair, all we looked at was yield and purity. We didn't even look at activity, okay? So some of those purified proteins that we had could actually be inactive, okay? So we didn't talk about activity. I didn't demonstrate that decreasing over time. I want you to think about another term called specific activity, and that's activity divided by total amount of protein. So what happens on a purification scheme to the total amount of protein from the beginning to the end? Does it increase, stay the same, or decrease? It decreases, okay? That total protein does, because why does it decrease? What are we getting rid of? We're getting rid of contaminants, okay? So as we get rid of contaminants, the denominator gets smaller, right? And hopefully our activity remains the same. So hopefully specific activity will increase over time. And I say hopefully because if you compare the specific activity of two different purification schemes, one done by me and one done by you, the one that has the highest specific activity is probably the one that's the purest. The caveat to that is you're making the assumption that every protein that is present is 100% active. Okay? So I can take something that's 100% pure, and if I boil it and inactivate it, guess what? The specific activity number is going to decrease. So specific activity is a quick way of determining purity, but there's that one caveat to that. Okay? And so you'll get a chance to calculate that with information from Lab 4 and Lab 5 data. So I want to go through an analogy. I like to use cooking analogies because most of you have done some type of cooking, but for some reason you get in the lab and you see a protocol and you just go, oh my goodness, what do I do? And your common sense goes right out the door, okay? Sometimes I wonder if it even came in the door, okay? But a protocol, that's a fancy name for what? A recipe. It's just a recipe. And so here's a recipe. And my daughter's having her birthday and she wants 12 cupcakes because she's got 12 friends coming, and I don't know how that's gonna work out, because I don't have a house big enough for 12. And so I'm gonna do the cheap stuff, the gold yellow cake. One box makes 12 cupcakes. And as I read that, one of the things it says, preheat the oven to 350 degrees. Well, I can do that, so I turn my oven on to 350 degrees. Am I gonna do anything else? Am I gonna sit there and watch the oven heat up to 350 degrees? Probably not. I'm going to be doing two things. So when you come to the lab, you're going to be creating a crude extract, and while that crude extract is incubating, you're going to be making a column. You're doing two things at once. Okay? Grease pan generously and dust with flour. Sorry, I'm lazy. I'm single, and I don't have a big pantry, and I choose that way. And so I don't have flour, so what am I going to do instead? No, I could, and I have, but I won't. Pan spray or cupcake tins. Hey, you bet that's the easy way to go, okay? And so you see, did I meet what the need of the what was the protocol? What was the purpose for adding the grease and the dusting with flour? So they don't stick. I met a method so it wouldn't stick. I like this next one here. Beat 3 to 4 minutes and mean speed. I'm sorry, hand mix 300. I'm not doing this. I'm not. And what happens if I'm hand mixing and I get to 299 and stop? Is my experiment failed? What's the purpose of that step? To mix it good, so it's homogeneous. What do we mean in reference to cupcakes? What do you want to get out of? What do you want to get out of there when you add that milk and flour together? The lumps. You want to get rid of the lumps, okay? And so I don't have a hand mixer. I'm not going to hand mix, and I don't have a hand mixer, but hey man, I got a KitchenAid, so I'll use it. And obviously my protocol's different because I mainly use the KitchenAid for 2 minutes. Have I done something wrong? No. You've got to stop and think about, use some common sense. What's the purpose of this step? Scientifically, what's the purpose of this step, and are you doing it? If it tells you to vortex something, the purpose of vortexing is to what? Mix it. And if you're just trying to make sure it's homogeneous, you could stick it on the vortex and go zip for 2 seconds, and it's mixed. I've seen students sit there 30 seconds to try to mix an enzyme. You know? Okay, come on. All right, and then I like this bake, you know, and it's got all these different things, and guess what? I got to figure out you're given a general guidelines. Which one do I do? Oh no! Well, you got to depend on your guideline, okay? And so as you come into the lab, you laugh at this, but everybody, there are 220 students in the course. Everybody gets the same recipe. I mean, you can't say that. Protocol. Gotta be scientific. Everybody gets the same protocol. Do you think everybody's going to make the same cupcakes? No. There's going to be problems. And you're saying, what did I do wrong? And a lot of times, I don't know. Okay? It's just little. Sometimes there's an art to some of the things that we do. All right. So throughout the experiment, you're going to experience some things like lost yield. Okay? That's always going to happen. You're going to have lost yield due to poor technique. All right? I see students that leave stuff in their centrifuge tubes, like the bowl on the right. What is that? And I know some of you say, that's a golden opportunity to get an extra lick. You know, I do that too, but that's lost yield due to poor technique. Oh, there you go. Differences between scientists. Who did the one on the left and who did the one on the right? You're also going to experience more loss in the process. Something's going to happen. Okay? And there's always experimental variances with the equipment. So what happens in the protocols, the recipes, and it didn't say this in this one, but a lot of times they say a good cook is going to turn the pan halfway through the cooking process. Why? Because the oven heats uneven, okay? And here's the problem. My oven doesn't heat correct. So that's obviously there's quality control issues there for cupcake making, or maybe that's lost activity. Okay? And so now you're down to icing, and there's more lost yield. That's always going to happen. And then you're going to have some more quality control issues Because God forbid I cannot give those 2 cupcakes to little girls because the sprinkles will go everywhere. They'll be everywhere. So I only have 4 cupcakes, and I got 12 girls coming to. What do I do? I lost all that yield. What do I do? I'm sorry.
Make some more?
Yeah, I get another box. You know, I buy 3 boxes, and because obviously I'm supposed to make 12 per box, and I'm only getting 4. I got my yields really low. Okay, so I have to make 3 boxes. You laugh at this analogy, but this will help you for one of your lab report questions. All right. So, with that in mind, hopefully these analogies, they're there to make you laugh, but they're also there to make you think. Okay? Make you think about applying what you do know to something new. And so most of you know cooking, and that's why I use those analogies. Going once, going twice. All right, very good, all of those. Increase the induction time. So that means that you're going from a G3 to maybe a G4, okay? Use a 30 mil pellet instead of a 15 mil pellet. The analogy is I only made 4 cupcakes, I need 12, just use triple my recipe, okay? Improve the braking efficiency. Maybe I shouldn't triple my recipe. Keep the original recipe and slap hands as I reach in to grab those cupcakes. Make sure that I'm efficient in transferring things from one bowl to the next, etc. Okay? So you're going to need to use those terms in class when you come to class this week. So for the remainder, we're going to kind of wake up a little bit, and we're going to start looking at the science behind what we're doing. And so there are several types of columns that we utilize in biology. In biochemistry, the first is gel filtration, also known as size exclusion. These are all different specific separation principles that you should understand. And size exclusion separates based upon size. And as something gets larger, intrinsically, its molecular weight is higher. So we're separating by molecular weight. Ion exchangers. We're not going to work with them in this course. There's some background reading on that on there. But these are charge-charge interactions. Cation exchangers bind positively charged proteins. Anion exchangers bind negatively charged proteins. We change the pH, we can change the proteinization state of the protein and have it bind or unbind particular exchanges. We're not going to go through those. Affinity chromatography separates based upon specific molecular binding properties. Similar to a lock and key, and I put the question mark there because we typically use that analogy of a lock and key for enzyme-substrate interactions. Okay, so that's why I put the question mark there. I want you to realize but it's a very specific interaction. Hydrophobic interaction chromatography, not done very often in biology, but it is a potential column separation based upon hydrophobicity or oily patches, and partition chromatography. Partition chromatography a lot of times is used in organic chemistry, not in biochemistry, because of our proteins will fall apart, won't behave properly under certain conditions. And so you're responsible for understanding size exclusion chromatography, and you're responsible for affinity chromatography. And so these are, we're going to do affinity chromatography in the lab. We'll talk about it, size exclusion here. And here we have these inert porous beads. You have these beads of gray on the slide, and they have holes in them. You could think of them as little sponges, okay? That have pores in there. And you have some large proteins and small proteins. Sometimes that says stable protein complex. So stable protein complex, primary structure, that's amino acid sequence. Secondary structure, what is that? What are examples of secondary structure? Data sheets and alpha helices. Those are secondary structures. Tertiary structure, what's tertiary structure? How they're folded, okay? And quaternary structure. How they're folded and interact together. This is a quaternary structure. And so if that quaternary structure is stable under our conditions, then we behave as one unit. So stable protein complexes. Okay, so tertiary structures referring to a single protein, and quaternary structures talking about multiple polypeptides or multiple proteins interacting together. So it turns out that these large molecules, these large protein or protein complexes, flow around the porous beads because they're just so big. Their size is excluded from the pores. That's why we sometimes call this size exclusion chromatography. Larger molecules are excluded from the pore. Where small proteins or small stable protein complexes flow in the pores of the beads, and they also go around the beads. So we know the quickest distance from point A to point B is a straight line. In the case of the large beads, they're not going through a straight line, but they're not getting stuck in the pores. And so they're going to be the first ones to elute. So the large proteins or stable protein complexes elute, come off the column first. So here's a kind of a time-wise schematic looking at a column, a single column, and looking at it at 5 different time points throughout time. And we have this column. The column is the gray material there. And that's that solid matrix. That solid matrix is the bed volume. That's the bed volume is that matrix, the volume of that matrix when it's ready to be used, and in our case, under gravity. And we have some type of porous plug to prevent things. It's in our case, it's going to be a Luer lock. And we're going to apply our sample on there. And over time, you're going to see that there's a delay before our sample comes off the column. The analogy is, I'm in a car, I'm driving, and I'm in Seattle where they're doing all the Google testing for the cars, the driverless cars. And I want to get over there, and there's an intersection there at that computer, and I got all these driverless cars right here. As soon as that intersection opens up, right, I can put on the gas pedal and go. As soon as but do I go through the column immediately? Do I go through that intersection immediately? What do I have to wait for? All these driverless cars to get out of the way, and they're going to go through that intersection also. You experience it every day when you go home. There's a delay. The intersection opens up. The Luer lock opens up. But there's a delay between you getting through that Luer lock or through that intersection. You've got to get those other empty-headed people out in front of you. Okay? And I say that because the important thing is the sample that was applied. All those cars that were in front, those driverless cars, it's just buffer. Okay? It's just, we call that the void volume. It has no proteins in it, no impurities. It's just buffer. Okay? And there's that delay there. So if we look at protein elution coming off of this type of column, on the y-axis you see protein concentration. I'm not telling you how they determine that. And on the x-axis you see the total volume, the elution coming off the column. And you can see the void volume. The void volume is listed right there. And the first protein to come off, size exclusion. They're being excluded from the beads. So in this case, the first protein is a large protein. The last protein to come off, 30,000 Daltons. The reason it was last to come off is because it weaved in and out, not only between the beads, but inside the pores of the beads. And so it had a longer distance to go until it got out of that Luer lock. So that's a size exclusion chromatography. You're responsible for that for future issues, okay? And so let's take a look at this. This is kind of a definitional thing. Definitional in nature. The void volume contains what? What does the void volume contain? Got a few more people need to click in. Remember, you get 1 point for clicking in and 1 point for clicking in correctly. All right, going once, going twice. Sold. Okay, there you go. All right, pretty good. Only buffer. There's only buffer in the void volume. Your crude extract or your sample that you applied is not part of that void volume. It's the empty space. It's the buffer that surrounds the beads in your column. So of these types of chromatography that we're going to be doing, we're responsible for gel filtration and affinity chromatography. We're going to be doing affinity chromatography in the lab today. This week. So this is nickel-agarose affinity chromatography, also known as the QIAGEN column. That's because it's patented. So if I said, hey, I know such and such, and you say, well, how do you know that? I said, I Googled it. You know immediately what I did, okay? So if somebody says, hey, how do you purify that protein? You say, oh, I use a QIAGEN column. You immediately know it was His6 tag. When you say, oh, I use nickel-agarose. Oh, you immediately know it was a His6 tag protein. Or you say, oh, I used a His6 tag. Immediately know that what was going on, okay? And so in this system, there's an inert matrix. In our case, it's agarose. There are different forms of that inert matrix. Cepidex, Cepacryl. They're used in different types of flow apparatuses. But we're going to use agarose as our inert matrix. And across that, there's a chemical linker. And that chemical linker, it is got three attachment points there, these 3 carboxyl groups, and it's also got this nitrogen. So it's kind of like there's this hand sticking out, a 3-fingered hand, and that 3-fingered hand, along with the palm, or the nitrogen, can coordinate with this nickel ion. And so you see that nickel ion is being held in place. You'll notice those bonds are dashed lines. This is not a covalent bond. The rest of this is covalently bonded. When I get the agarose matrix with the chemical linker by itself, it's white. When I add the nickel to it, which is a metal ion, that's why sometimes we call this immobilized metal affinity chromatography, because we immobilize a metal ion there, it turns blue. So your column is going to look blue under normal lighting because it's holding that nickel ion. That nickel prefers to be coordinated with 6 other ions. In this setup, it's only being coordinated by 4. The other 2 that it coordinates with is the nitrogens out of the histine ring. Okay? And so, if we look at a column, a bigger version of this, we're just looking at the lower version of this. Let's look at a bigger version of this. On the right is the column itself. There's our target protein, GFP, 3-dimensional structure. And at the end terminus, we have 6 histidines in a row. 6 histidines are more efficient than 2 histidines in binding to the column. That's just been done empirically. And hence, we use a His6 tag. I've seen people use a His8 tag before. It works also, but it requires, it's a little bit harder to get your protein of interest off the column. And so most people will just use a His6 tag. What you need to recognize also is that the His6 tag, in order for this to bind, we're thinking of the His6 tag in a primary sequence. Okay? If you have 6 histidines that are real close to each other in a tertiary sense, okay, then those types of proteins could also bind to the nickel-agarose column. So whenever you've got a multitude of histidines real close to each other in three-dimensional space that will readily bind to nickel augurs. And so we've got to loop that once we've bound that. So once we bind our protein of interest, if we keep washing the column, what's going to come off the column? So if I have this, I put my crude extract on, it binds, the GFP binds the column. If I keep washing it with buffer, will the His6 tag protein come off? No, but what does come off? All my contaminants. So most of my proteins should come off the column, and that's most of my total protein, and that should be the contaminants. But at some point in time, I want to get my protein of interest off, and that's when I got to elute it. And there are two methods for eluting off of a nickel augurous column. So I've drawn a line there to indicate there's a method on the left and a method on the right. The method on the left is changing the pH of the buffer. And when that happens, that changes the ionization state of the histidines, and as they become unionized, neutral charge, then those proteins will fall off the column, come off the column. It can be done, changing pH could have an effect on your protein of interest. We saw it with phosphatase enzymes. At a mechanical level, it's a little bit more difficult. It can be done. It's kind of like if I'm in a bakery shop and I'm putting out hundreds of donuts, hundreds of cupcakes a day, I'm probably not going to hand mix. I'll go ahead. I could, but I'm probably going to use that KitchenAid. I could use pH changes. It's just kind of a pain technically because you got to have multiple buffers now. Okay, and sometimes it's just easier to have one buffer. So that's one method. The other method is to compete off the His6 tag protein with a huge excess of something that looks like and smells like histidine. Okay, that is to say, something that has very similar chemical properties as histidine, and in this case, it's imidazole. So the more imidazole you add, those proteins that are bound to the column will unbind because of competition. So, I'm going to go through a couple series of slides to help you understand how we represent proteins. You're going to need this for a lab report question. On the left is a protein being represented in 3D ribbon diagram. That's a 3D structure, and in this case, it's being shown as a 3D ribbon diagram. The only way you can show that is if you've done a 3D crystal. So I'm showing you that GFP has been crystallized before. Okay? It's not been crystallized with that express His6 epitope tag system that I have. Okay? So that's why it's just kind of shown as a floppy line here, because we don't know what structure it is. The other way to represent a protein is through a schematic style diagram. And that's shown there at the bottom. And so on that diagram, I'm showing you important features of that schematic diagram for the purposes right now. I'm showing you the His6 tag region, and I'm showing you the RGFP-UV region. And I'm also showing you that by this color combination, the green portion is most likely the wild type, or the GFP-UV portion. These blue portions were added they were added at the genetic level, at the DNA level, and they're not part of the normal GFP. There's a nomenclature. You're reading left to right on these. The left-hand side is always the N-terminus. The right-hand side is always the C-terminus, or carboxy-terminus. Let me show you an analogy to this so that we can so that you can learn how to do this for GFP. So you don't need to memorize this, but there's some things to picture out of this. There's that three-dimensional structure of an enzyme T7 RNA polymerase. Okay, the polymerase that's in our expression system. And there's the amino acid sequence, or the schematic style diagram. How many amino acids are present in T7 RNA polymerase? 883, so that's how we're reading it. There's the carboxyl end. And so there's an amino acid there. And in this particular case, here, there's only really one domain. There's a domain plus a tag. The domain is the GFP domain, the Beta Barrel, it's a self-described domain. It's functional as a unit by itself. In the T7 RNA polymerase, if you look at the primary structure, that's what that looks like. But if you look at the three-dimensional structure, there are certain regions that we call domains that have functionality. And typically, sometimes a domain can be expressed and has its own activities separate of all the other domains. Not necessarily always the same, but so there's the internal domain, the thumb domain, the fingers. These are very specific to that. You don't need to know those, but what you do need to know is how to read that. I'm interested in you knowing how to read this kind of information. So the N-terminal domain. How many amino acids in the N-terminal domain? Well, it turns out that if you said 325, we're reading it wrong. Okay? The first amino acid of the N-terminal domain is right here, amino acid 1. The first amino acid of the thumb domain is amino acid number 325. So how many amino acids in the N-terminal domain? 324. Okay? Now, looking down here at the extended foot, or this little white area there, that 883rd amino acid, is it part of the next domain or the previous domain? Well, there's no next, so it's part of the previous. All right? So that's how you read that. Okay? So, here's an example of schematic style diagram of a protein, probably pretty similar to what you might be presenting in a lab report question. There's the N-terminus, there's a C-terminus, there's a C-terminal tag, it's got a His6 and a big epitope tag. It's got a very specific region within this transcription factor AG. Amino acids 331 and 341, including 341, the way that's diagrammed there, because I've underlined it. That means 331 all the way through and including 341 is a DNA binding domain region. Knowing that, knowing how we read these, let's take a look at that particular schematic style diagram, and I'd like you to answer this question. How many amino acids are present at the C-terminus of transcription factor AG? Another question I could ask you, and there's information on there, I'm not going to, is what is the estimated molecular weight of the transcription factor? Knowing how many amino acids are present and the average molecular weight of the amino acids. And you'll get that opportunity to answer in the future. Not today, but sometime in the future.
What's wrong? What did you get? It's not working. No.
This class over 349? Oh, I didn't know that. Excuse me. I've never thought one that. I'm gonna skip through. If you want to learn that, there's 364. There it goes to 380. If you count those 1 through 17, so mathematically it's minus and plus. This stuff is just how you're doing it this week. I will post these on OO2 section, these last 2 slides.
No, it's because like he